Good afternoon, everyone. Today we will be starting with the session 3 tutorial where we will kind of summarize the week 3 content and then look at few of the question answers, sample questions and answers. So in this week we have things based on what are the components of the week's content will have what are the components of cell and how um, the cellular components, cellular organelles are organized and what are their features, etc. Okay, so we'll start with the session. So when we talk about eukaryotic and prokaryotic cells, um, so here, when we talk about the difference between prokaryotes and eukaryotes, so in prokaryotic cell lacks membrane-bound organelles, though they may have simpler structures like ribosomes, which are actually protein synthesis factories. Whereas in eukaryotic cells, the membrane-bound organelles are present like mitochondria, ER or endoplasmic reticulum, Golgi apparatus, lysosomes, and many more. In prokaryotes, true nucleus is absent. Genetic material is present in the nucleoid, which is not really enclosed by a membrane. And eukaryotic cells have a true nucleus enclosed by nuclear membrane containing genetic material. Prokaryotes, the size of the cell is smaller, and in comparison, the eukaryotic cells are bigger in size. Prokaryotes have single circular chromosomal DNA. It may also contain plasmids. Um, and eukaryotic cells have multiple linear chromosomes or DNA in the nucleus. And prokaryotes have unicellular structure, whereas eukaryotes can be unicellular or multicellular. Like yeasts are there, which are unicellular or monocellular eukaryotes. And then in prokaryotes, lysosomes and peroxisomes, which are again um, cell organelles, we will look at it. We'll look at the features. And however, in prokaryotes, these are absent, whereas in eukaryotes, both these organelles are present. Microtubules are absent in prokaryotes, whereas microtubules are present in eukaryotes. And in ER, again, we looked at it. ER, mitochondria, etc. are absent in prokaryotes, whereas in eukaryotes, both of these things are present. And cytoskeleton is absent in prokaryotic cells, whereas cytoskeleton is present. Ribosomes are smaller in prokaryotic cells. Ribosomes in eukaryotic cells are larger. Vesicles are present in prokaryotic cells, whereas vesicles are present in eukaryotic cells as well. Cell wall is present in prokaryotes. Cell wall is not really present in cell wall is present in eukaryotes actually, in and plant cells as well as in fungi. However, some may or may not have the cell walls like the animal cells. Okay, and prokaryotic cells divide or reproduce primarily through binary fission or asexual reproduction. They do show sexual form of reproduction as well, where some form of genetic material gets exchanged. Whereas in eukaryotic cells, they reproduce through mitosis, asexual or meiosis or sexual processes. And the structure and function of a prokaryotic cell is generally simpler, whereas eukaryotic cells are more complex structurally and functionally due to the organelles because they contain multiple organelles and cellular compartmentalization. Okay. So next, if we look at the structure of just a minute. um, why is- Let it be. Okay. And when we talk about the structure of a prokaryotic cell, um, so when we talk about the structure of a prokaryotic cell, what is happening here is this is the cell. Now there are multiple outer cell wall layers. Okay, I mean cellular layers. Cell wall is there. Then, like even before the plasma membrane or the outermost boundary of a cell comes into picture, the plasma membrane. Before that as well, the plasma membrane is also covered by multiple cellular layers. One of them is the outer. So if we start from the outermost part, it is the flagella or outer flagella, this part here. So a flagellum is attached to the bacterial capsule. If this is the capsule, um, which is a central feature of most of the prokaryotic cells, especially those bacteria which are motile or can move through the move through certain media. It provides motion or locomotion to the bacteria and is responsible for chemotaxis of bacterial cells. Chemotaxis means their movement in response to some kind of chemicals in their surrounding or in their environment. And movement of bacteria towards a chemical gradient such as glucose is known as chemotaxis. So flagellum is a part of the cell wall and its motion is regulated by motor proteins present inside the cell. And flagellar motion is an energy consuming process and it is governed by ATPase present at the bottom of the shaft. Okay, like at the this is present at the base of this flagellum, and it is made up of the protein flagellin, and reduction or suppression of flagellar protein reduces bacterial infectivity or pathogenicity and its ability to grow. Okay, and we'll just check once if anyone else wants to. So, hi Rashmi. Actually, today I just started the session now. So we are looking at the different types of cell organelles and what are their features and how things are organized. So this is the first slide only where we have started looking at the prokaryotic cells. Okay. Hopefully my presentation is visible to you. Mm, yeah. So, outer flagellum, yeah, we looked at the flagellar, and then there are bacterial surface layers present where there are the cell possesses three anatomical barriers to protect the cells from external damage. So there is this capsule, the outermost part, and it is made up of high molecular weight polysaccharides, and it is impermeable to water or any other aqueous solvent, whereas it is also even responsible for giving antigenicity of bacterial cells. Okay, and cell walls in bacteria is also, and its response to gram staining is the basis of classification of bacterial species. Okay. Now, if we talk about the cell wall, now there are certain cells which can take up a certain stain, which are called or take up as well as retain that particular stain, which is gram positive, as well as there are certain bacterial cells which do not take up the particular stain. It is called gram negative bacterial cell wall. Okay. And um, And cell wall, so if we talk about the cell wall composition is in gram negative and gram positive bacteria, it is different, and which eventually gives it the ability to retain the gram positive, I mean gram stain or not. Okay. So cell wall composition is in gram positive bacteria. Um, there's a peptidoglycan layer as well, which is thicker, like much thicker in gram positive bacterial cell wall, whereas in it is actually thicker, I mean very thin in gram negative cell walls. Okay, gram like cell walls of gram negative bacteria. So if we talk about peptidoglycan, it is actually a polymer of NAG, N-acetyl glucosamine, and NAM, N-acetyl muramic glucosamine, which is linked by beta-1,4 linkage. So sugar polymers are attached to the peptide chain composed of amino acids L-alanine, D-glutamic acid, L-lysine, and D-alanine. And peptide chain present in one layer is actually cross-linked. These are the cross-links here. Okay, these ones, right? These are actually cross-linked to the next layer to form a meshwork and be responsible for the physical strength of cell wall. Peptidoglycan synthesis is targeted by antibiotics such as penicillin, whereas lysozyme present in human saliva or tears degrades the peptidoglycan layer by cleaving glycosidic bond connecting NAG-NAM to form polymer. Okay, so if we talk about gram negative cell wall, you see this is the cytoplasm. Now this is the plasma membrane, inner membrane and outer membrane. Then there's peptidoglycan layer, very thinner peptidoglycan layer, and then there's outer membrane having lipopolysaccharides, porine proteins. Okay, there are lipoproteins sticking in between as well. Whereas in gram positive cell wall, this is the cytoplasm, this is the cell membrane. All right, and um, then it's a thicker layer of peptidoglycan, and then there's lipoteichoic acid, which is present in gram positive cell wall. So, uh, Rashmi, uh, is my presentation visible to you? You can write it in the chat also if it's okay with you. Because hopefully it should be visible, but still. Next, so if we look at gram staining, so what happened? There was um, particular stain developed by a person named Gram. So what the person did was, if he saw that when he applied crystal violet, a purple colored dye onto cells, bacterial cells, then he applied iodine or a mordant which can actually fix that particular stain. And after giving alcohol wash, it leads to decolorization. And further, when another counter stain is applied, saffronin, then some of the cells get stained with saffronin, whereas the previous cells and some of the cells are colored purple only, the very first crystal violet stain. Okay, so based on this, those cells which could retain this purple colored crystal violet dye were called as gram positive cells, and the ones which could not really retain the dye even after alcohol wash and then got counter stained with saffronin were called as gram negative cells. Okay, so what is happening here if we look at it carefully? So gram positive and gram negative, so both the cell walls affix the dye. Okay, the very first one that is being used to stain the cells. So in gram positive cells, what is happening? There is a thicker layer of peptidoglycan, right? So the dye complex is trapped inside the walls, whereas like in case of the gram negative cells, it has a very thin layer of peptidoglycan, right? So there is no effect of iodine, which can affix the dye or gets like properly, hold the dye together. And even after washing with alcohol, what happens here in gram positive cells is that the crystals of the very first dye, the purple dye, remains in the cell wall. However, in case of gram negative cells, what happens? The outer membrane gets weakened and the cell wall loses the dye. And now when the counter stain, which is saffronin here, is actually getting masked by the violet purple, like darker violet purple dye, whereas Here, since there's no purple colored dye, the alcoholic wash loses the purple dye, and hence it gets stained with saffronin, and we can see red dye or pinkish dye, like pink colored cells on the, like under the microscope. Okay. So this is the same slide almost, where the gram-positive bacterial and gram-negative bacterial cell walls have been compared. Cell membrane, peptidoglycan, thinner peptidoglycan, then lipoprotein. Then there are lipopolysaccharides and stuff in gram-negative bacterial cell walls. Okay. Whereas in gram-positive one, we see a very thick peptidoglycan layer. Now, if we talk about peptidoglycan layer, so peptidoglycan is actually a rigid mesh made up of rope-like linear polysaccharide chains. These are the repeating units NAG, N-acetyl glucosamine, and NAM, N-acetyl muramic acid. Okay. And these two entities, moieties, are actually connected by beta 1,4 glycosidic linkage. All right. And yeah, and then there's branching and stuff, which we see here. This part here, all these are branches which are actually leading to this cross branching, cross linking leads to a very thick mesh of peptidoglycan layer and a thicker cell wall in gram positive bacteria. Okay. Now, in we see that in this bacterial cell or prokaryotic cell, we see some invagination here, which is called the mesosome. Okay. So, mesosomes are actually the folded invaginations or extensions of the plasma membrane inside the cytosol or cytoplasm of the prokaryotic cells. So, mesosomes are still being studied extensively, and what is its role? So, it is basically a cellular compartmentalization and organization. It provides an additional surface area within the cell here, creating compartments that allow for the spatial segregation of different cellular components. It helps in cell division and DNA replication, plays a role in the formation of the septum or wall during cell division by helping in invagination of the plasma membrane, and helps ensure the proper distribution of genetic material during cell division. It has also role in respiratory processes. It provides an increased surface area for the attachment of respiratory enzymes like cytochromes and ATP synthase. It has been found to have role in protein secretion and localization, where it helps in the transport of newly synthesized protein, facilitating their localization to specific cellular compartments or external secretion. Even it helps in cell wall synthesis. It participates in transport and deposition of peptidoglycan. Now, when we talk about cytosol and other organelles in a prokaryotic cell, so prokaryotic cells do not have any membrane-bound organelle, and even if it's present, the organelles present are ribosomes or 70S ribosomes, genetic material, whereas electron transport complexes are embedded within the plasma membrane. Because mitochondria, those are not present here. And chromosome and extra chromosomal DNA are found in prokaryotic cells. So the genetic material is mostly in the form of circular DNA known as bacterial chromosome. So this particular chromosome contains the genetic elements for replication, transcription, and translation. Bacterial chromosome follows a rolling circle mode of DNA replication. The genes present on the chromosome does not contain um, introns, or introns are basically like uh, if there is maybe introns are basically like continuous interference, like when we have transcribed RNA. So when the RNA is transcribed from the DNA, what happens there is both coding as well as non-coding sequences are transcribed, and so the coding ones are called exons, and in between, especially this is the case in case of eukaryotes, where the exons are interfered in between by interfering sequences called introns. However, in case of prokaryotes, that is not the case. The non-coding regions are not really present, and it's a polycistronic gene in case of prokaryotes, and it gets co-translated to proteins, even because there is no division between proper compartmentalization between the nucleus and the cytoplasm in case of prokaryotes. Unlike in eukaryotes, you have the RNA getting synthesized inside the nucleus, RNA or mRNA getting transported out of the nucleus, and then protein synthesis getting like taking place in the cytosol, right? By ribosomes. But however, in case of prokaryotes, that is not the case. In case of prokaryotes, we see transcription like RNA synthesis and protein synthesis translation being coupled. Like from the same mRNA, the mRNA getting synthesized by RNA polymerases, but the same mRNA that is getting synthesized is bound by ribosomes, and proteins are getting synthesized simultaneously. So this is called transcription translation coupling that we see in case of bacterial cells or prokaryotes. Okay, this is not the case in eukaryotic cells. All right, and so when we talk about bacterial plasmids, so plasmids are widely used for cloning of foreign DNA into the bacteria as a host strain, and even so these bacterial plasmids even carry certain antibiotic resistance genes. So whenever these bacterial plasmids are used as vectors and they get inside some other cells, then that particular antibiotic resistance get the property of antibiotic resistance gets inside the other cell type, and eventually those cells can actually harbor and show antibiotic resistance. Okay, now if we talk about the different types of plasmids, plasmids are basically double stranded circular DNA, and that is the genetic material in bacterial cells, right? So if we talk about the different forms of plasmids, so bacterial plasmid is a double stranded circular DNA which can exist in predominantly three forms. Okay, so one is this supercoiled DNA, one is this covalently closed circular DNA, and one is open circular DNA. So what happens is, if both the strands of circular double strands are intact, then it is a covalently closed circle. Okay. Whereas if one of the strand has a nick, then it acquires the conformation of open circular DNA. Nick as in it is a double stranded one. Nick is where the phosphate backbone, like we don't, the phosphate phosphoesterase linkage is not there, phosphodiester linkage is cleaved, and hence you don't have this particular, I mean not both the strands. If both the strands have a nucleotide altogether missing, then it's a gap. However, when we call it a nick, during nick this linkage, one of the bonds is broken here, and hence we call it as open circular DNA. However, many times supercoiled DNA, this is the predominant like intact plasmids, like circular plasmids can show supercoiling. Okay, these covalently closed circular DNA can only uh, turn upon itself and as a result acquire a supercoiled configuration which is extremely compact and actually runs really fast on a on an agarose gel. Agarose gel is basically where you actually load your sample, like that is a test of seeing where do you you actually purify plasmids, you actually take out chromosomal or genomic DNA from organisms, etc., and then you run it on agarose gel. Agarose is a matrix of polysaccharides. Okay, and so through that matrix having different kinds of pores in between, through those pores these biomolecules can actually, or macromolecules can actually pass through those pores depending on what is their size. So the really larger sized plasmids, suppose if we are talking about plasmids here, so those really larger sized plasmids would be having difficulty to pass through the pores, whereas really smaller ones can actually pass through the pores very easily. Okay, so based on this, their mobility on such cross-linked matrix, we see we can actually differentiate and even check the quality of the genomic DNA or plasmid DNA that have been isolated. Okay, so this is again the same thing, circular DNA and covalently closed circular DNA, double-stranded DNA, nicked circular double-stranded DNA. Only the linkage has been broken here, right? We don't have the ester linkage here. Whereas in case of gapped circular double-stranded DNA, we have one complete nucleotide missing from here. Okay, one nucleotide or maybe more than one. At least there is a missing nucleotide here. Okay. Now there are some minimum molecular components that are now bacterial plasmids are actually very commonly used and regularly used in case of genetic engineering and uh, cloning. Cloning is some wherever we want to insert any gene of our interest in a foreign organism or foreign cell. We use bacterial plasmids as vectors or carriers of that particular gene. Okay. So for that, definitely it needs to have origin of replication so that these plasmids, when they are inserted into a foreign cell or foreign organism, they can form like self-replicating entities. So it needs to have an origin of replication from where the DNA can be replicated, right? And then it should have a selection marker. Mostly they possess, these plasmids possess antibiotic resistance genes. So when, if this particular plasmid gets inside a foreign cell, suppose, and how do we really know that the foreign this particular DNA has been taken up by a cell? So based on the character, the cell initially like inherently doesn't really poses resistance to suppose an antibiotic kanamycin. Okay, however, the plasmid DNA that we are introducing into the cell has resistance to shows resistance to kanamycin. Okay, has the gene that shows resistance to kanamycin. So if that particular plasmid DNA showing kanamycin resistance with the kanamycin resistant gene inside it gets inside the cell, then eventually the cell which did not have the property to show resistance towards kanamycin, it was the cell could not really grow in a kanamycin containing medium. Now, if the plasmid containing kanamycin resistance gene is inserted into the cell, then eventually the cell, if the cell has taken up that particular plasmid, then that particular cell can easily thrive in a kanamycin containing medium. So we can actually differentiate between the cells which have taken up that plasmid and the cells which could not really take up the plasmid based on the medium in which they are grown. Okay, where we put in the antibiotic. So if the cells are growing in that antibiotic, that means it has taken up the plasmid, whereas the cells, when if they aren't able to grow on the antibiotic containing medium, then those cells aren't really haven't really taken up the foreign DNA or this plasmid DNA. Okay. So this is one of the very first plasmid to be synthesized, having one origin of replication here and resistance to two different antibiotics. Okay, and this is another regularly used experiment, like a plasmid in the laboratories. Okay, and now if we talk about plasmid DNA extraction, so what happens is we have to culture the cells. Definitely, suppose if we are talking about bacterial cells, E. coli cells. Okay, so plasmids are present inside the cells. So the very first thing that one has to do is culture, grow as many cells as you want, right? So we have to have many copies of the same cell that have taken up the plasmid. And then we have to break the cells open, which is the lysis method here. Okay. So upon breaking the cell open, because these bacterial cells have, cell wall and stuff, then plasma membrane also has to be disintegrated so that the components inside, the cytosolic components and the different um, like plasmids or genetic material, etc., or ribosomes, etc., can come out, right? So after lysing or breaking the cells open, uh, using multiple types of specific buffers, eventually what we do is we take out the genetic material, and that can also have its chromosomal DNA, which is really heavy and large. Whereas it can also, like along with that chromosomal and like genomic DNA, one can have these lighter double-stranded circular DNA called plasmids as well. Now, for plasmid DNA isolation, multiple rounds of chloroform and phenol, etc., are given. Treatment is given so that finally we can purify the plasmid. Okay, so we'll look at it one by one slowly. So in plasmid DNA extraction, it is trickier because plasmid DNA must be separated from the genomic DNA, which is simplified as gDNA, to avoid cross-contamination. Genomic DNA or chromosomal DNA is much, much heavier than the plasmid DNA or circular double-stranded DNA that are present. So the very first thing is breaking the cell open, which is lysis here. So lysis is more subtle than simply digesting the cell wall with enzymes, or beating it with glass beads. Like we are mechanically trying to or physically trying to break the cells open by giving it pressure using glass beads. Okay, which is called bead beating method kind of. Okay. So usually the component that definitely should be there in case of lysis buffer is sodium hydroxide, strong alkali, and detergent SDS. SDS is sodium rhodecyl sulfate, which is detergent, which again helps in breaking the cells open. Okay. So what also happens is this treatment of detergent and alkali completely denature the plasmid as well as genomic DNA, the chromosomal DNA. Okay. So denaturation means DNA is double stranded, right? So by treating it with strong alkali and detergent, what happens is the two strands of DNA is actually held together by hydrogen bonds, right? So, so those the hydrogen bonds between those two strands are actually separated, broken, and the DNA gets separated into single strands. It is really crucial. This step is crucial to perform quickly, because in case we are denaturing it excessively, that may result in an irreversibly denatured plasmid, right? Like not only the hydrogen bonds between the two strands get broken, but it can irreversibly denature some bases or something, where you don't get to separate it out. Okay. Whereas, just a minute. So next what we have here is, first is lysis. So lysis is more subtle, where we are treating it with alkali and detergent. Then we have neutralization step, where the main thing is potassium acetate, which eventually is to neutralize the overall treatment that was given using strong alkali and detergent. So potassium acetate helps to renature the plasmid, and this is a very crucial step to actually separate the plasmid as well as the genomic DNA. Okay. So if we talk about the plasmid and genomic DNA, so genomic DNA, plasmids are really small and it can easily reanneal, forming double-stranded DNA, whereas genomic DNA is too large to reanneal fully, and it tends to tangle the double-stranded, like the genomic DNA or chromosomal DNA, so that the complementary strands actually remain separated. So what happens is, when we centrifuge it, the genomic DNA, which is bound to protein, forms a pellet, while plasmid DNA remains in the supernatant or in the soluble form. So ideally, we should not really vortex or mix the sample very vigorously, because chromosomal DNA can break in between and eventually give rise to smaller double-stranded forms, which can come into the soluble fraction and lead to plasmid DNA contamination or genomic DNA contamination. Okay. So broken genomic DNA may be small enough to reanneal and go into the solution with the plasmid. Okay. Next, Next is purification. So the plasmid DNA in the supernatant can then be precipitated with ethanol or cleaned up using phenol chloroform or a spin filter. Okay. So basically, mostly it's with ethanol, where what happens is it has sodium acetate and ethanol, where ethanol, so water has a higher dielectric constant. So if we talk about the Coulombic forces, the charged DNA is negatively charged and water is a polar solvent, right? So, so what happens here is that sodium acetate salt is there. So DNA is coated with positively charged sodium. Now, for these two charged entities to really come near each other, what happens is the DNA, I mean the ethanol present in the medium, if we add ethanol, the dielectric constant of ethanol is comparatively lesser than water. Now we know that the Coulombic force of attraction between two charged species is like inversely proportional to the dielectric constant. So if water's dielectric constant is, if I remember correctly, is around 80 or something. So if it's too high like that, then um, one it is problematic for the, if it's too high, then the Coulombic force between the two charged species is comparatively less. So to decrease the dielectric constant and increase the attractive forces between the Coulombic forces between two charged species, the DNA and the sodium ions in this case, negatively charged DNA and positively charged sodium. So this ethanol is added so that these two species can come together, and the positively charged sodium coats the negatively charged DNA molecule, and this coating eventually helps um in kind of interfering, it interferes with the um access of water molecules onto the DNA. Okay, so eventually the interaction of DNA with the water molecules with the polar aqueous environment around it gets interfered, and eventually the DNA precipitates out. Okay, so that is what this particular principle is taken care of in case of precipitation of DNA. Okay. So if we talk about, I was talking about, um, yeah. So, so when we run it on an agarose gel, the plasmid DNA can have multiple forms. One is the nicked form, we looked at it. One is the linear form where both the strands get nicked. Okay, and we can have supercoiled form where the complete closed circular form coils upon itself. So the supercoiled DNA is the native DNA conformation found in vivo and occurs when extra twists are introduced into the double helical strand, plasmid DNA strand. We are talking about plasmids here. So in case of DNA plasmid preps, this superhelical tension cannot be relieved because the ends of the plasmids are joined together, and supercoiled DNA migrates faster than predicted in an agarose gel due to its conformation, because it's supercoiled, right? It's coiled upon itself. So overall it is a very compact ball-like structure kind of, if you look at the hydrodynamic radius. So it can easily pass through the matrix of the gel that we were talking about, agarose gel earlier. So it can easily pass through the pores and eventually run the fastest on an agarose gel matrix, on a porous matrix rather. Okay, and then there's supercoiled DNA. Then you have the DNA in the supercoiled form that is not easily accessed by the replication machinery. And during replication, cellular topoisomerases, these are again enzymes that nick one of the strand of the DNA helix and relax the superhelical tension, thereby allowing the polymerases to gain access to the DNA. Okay. And when we talk about the linear form of plasmid DNA, how it happens? Because the DNA helix, both the strands of the DNA double helix is actually cut and at the same place, and linear DNA generally migrates between the nicked circular form and the supercoiled form. However, it may also sometimes migrate at the same position as the nicked circular form. Okay. Now, if you usually get DNA in the linearized form, when hoping for the supercoiled form, this is the native conformation that we are talking about in plasmid preps. So it is due to nuclease contamination. Nuclease is nucleic acid digesting enzymes. Okay, so if there is some kind of nucleic acid digesting enzymes like DNases, then it can actually cut both the strands or at the same place, eventually linearizing the circular DNA. Okay, and alkaline lysis that we talked about using sodium hydroxide can that is to disrupt the hydrogen bonds by alkaline conditions, right? So if in case the alkaline lysis step is too harsh, that is suppose you are keeping it for too long with your plasmid sample, then the DNA can permanently denature, giving single stranded closed circles that migrate this one ahead of all other plasmid forms on the gel. Okay, gel or porous matrix, agarose porous matrix. Now, all this were pertaining to the prokaryotic cells. Okay. Now, if we talk about the eukaryotic cell, eukaryotic cells are much more complex, definitely, and contains many membrane-bound organelles to perform specific functions. It contains nuclei or nucleus isolated from the cytoplasm, like well compartmentalized nucleus is there, and it is also enclosed in a well-defined plasma membrane. Now, again, if we talk about eukaryotic cells, broadly, we can talk about plant cells and animal cells. So, in plant cells, cell wall is present. In animal cells, it's mostly absent. The size of plant cells are comparatively larger, whereas like than the animal cells. Chlorophyll is present in plant cells, whereas it's absent in animal cells. And vacuole, there's a large central vacuole in case of plants. Okay. Large central vacuole in case of plants, whereas in animal cells it's really small and there are many number of vacuoles. Mitochondria are few in number, whereas animal cells have more number of mitochondria. There's lysosome. Uh, it is present in plant cells, but comparatively, it's much lesser as compared to animal cells. There are glyoxysomes which are present in plant cells, however, it's absent in animal cells. Then there's cytokinesis. Cytokinesis means the division of the cytoplasm. Okay, and we will look into this particular thing in later slides where we talk about cell division. Okay, cell cycle and cell division. So this is the cytoplasm or cytosol. Cytoplasm is the area between the cell membrane and the nucleus containing organelles. And cytosol is the gelatin-like aqueous uh, material in the cytoplasm, which actually has salts, minerals, organic molecules, and even helps in the transport or like cross communication, cross talk between all these substances. Okay. So it is the liquid part, basically the cytosol, which is filled inside the cell and it contains water, salt, macromolecules like proteins, lipids, RNA. It has an array of microtubule fiber running throughout the cytosol to give vesicular structure to its destination. Besides this, cytosol exhibits salt to gel transition, and such transition actually regulates multiple biochemical and cellular processes. So if we talk about cytosol functions, it doesn't have a well-defined function. It serves as a medium to exchange the materials between organelles. It plays a role in several cellular processes, for example, signal transduction, translation. Okay. Now we talk about the nucleus. This is a eukaryotic nucleus that we are talking about. It has a well-defined compartment. The nucleus is bound by nuclear membrane. There are nuclear pores in between, and it has a nucleolus and chromatin, the genetic material spread all around. Okay, so nucleus is basically the storage body where the cell's hereditary material or the DNA is stored. Nucleus actually coordinates various important cellular activities like protein synthesis, cell division, growth, and a host of other important functions. So we talked about the chromatin, like these thread-like structures which is the genetic material, right? So this particular genetic material in question, so that chromatin can actually again have two different forms. One is the heterochromatin and one is the euchromatin. U means true chromatin. Okay. So heterochromatin DNA is generally not really active, and in interphase, so this is one of the earliest phase before the cell divides. So this the chromatin gets really compact. Okay, and heterochromatin, euchromatin is actually generally more active and it gets transcribed. Okay, so that can be heterochromatin is very densely packed, and it's not really accessible for the transcription machinery. Transcription machinery is the one like RNA polymerases and other transcription factors which can actually read the genetic sequence on DNA and encode RNA. Okay, transcribe RNA, synthesize RNA from that from the DNA sequences. However, that is possible in case of euchromatin, whereas heterochromatin, since the DNA is densely packed, so the sequences, individual sequences, nucleotide sequences on the DNA is not really readable. Okay. Now, when we talk about the nucleus, the eukaryotic nucleus has a double layer of membrane known as nuclear envelope. Outer membrane of nuclear envelope is continuous with rough endoplasmic reticulum and has ribosomes attached to it. And the space between two membranes is perinuclear space. And nuclear envelope is often, envelope often has a nuclear pore. We looked at the nuclear pore here, these ones. Okay, these are the nuclear pores in between. So, if we see an average nucleus has approximately 3000 to 4000 pores per nuclear envelope. Now, if we talk about the nuclear pore dimensions, it is approximately 100 nanometer in diameter and consists of different kinds of proteins. Basically, it's a pathway for transfer or gateway for transfer of material between the nucleus and the cytosol, like inside the nucleus and outside the nucleus. RNA formed after transcription from DNA within the nucleus has to move out, right? And from that RNA only, that RNA will be read and encoded for like translated into proteins, right? So that RNA has to come out of the nucleus where, so the reading machinery that is actually the protein synthesis machinery, which are the ribosomes, that particular those factories, the protein synthesis factories are actually present in the cytoplasm. It's not really present in the nucleus. So the RNA that is synthesized inside the nucleus has to come outside to so that it gets translated into proteins. So, if we talk about the nuclear pore complexes, these are actually channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. Okay. So, if we talk about this is inside the nucleus, this is the inner nuclear membrane, and that is the outer nuclear membrane. This part here is the cytosol. Now, depending on their size, on the sizes of the molecules, they can travel through the nuclear pore complex by one of the two mechanisms. One is the passive transport based simply on the concentration gradient, and one is the active transport, which is energy dependent. Okay. Then we have, this is a mitochondria, another membrane-bound cellular organelle, okay, present in eukaryotes. And again, mitochondria have two membranes, the outer membrane and the inner membrane has like foldings in between called cristae. And even mitochondria has its own ribosomes, and it is the powerhouse of the cell ATP generating machinery, right? So the ATP generation is based by mitochondria is based on chemiosmotic theory. It actually explains how ATP is generated in the mitochondria. This hypothesis is that most ATP synthesis in the respiring cells comes from electrochemical gradient across the inner membranes of mitochondria. Inner membranes means these ones, the ones that is folded into cristae, the pink, the edges of the of this pink area. Okay. And there are NADH and FADH2 molecules. The energy of these molecules, while forming these molecules, during the oxidative breakdown of energy-rich molecules like glucose, are used to actually drive the synthesis of ATP. And this is driven by the proton motive force, which is developed due to the proton gradient or H plus ion gradient, and then the subsequent movement of H plus ions across the membrane, across the mitochondrial membranes, which eventually provides energy required for ATP synthesis. So this is what we were talking about. So this is the outer mitochondrial membrane, that is the cytoplasm. Then there is some space, periplasmic space between the two membranes, inner mitochondrial membrane and outer mitochondrial membrane. And the inner mitochondrial membrane has these complexes which are part of the electron transport chain, these green ones. Okay, so these are actually four enzyme complexes here, the green ones that we see. So these four complexes are part of the electron transport chain. This is embedded in the inner mitochondrial membrane. Okay, so the very first is hydrogens from NADH and FADH2 are separated from protons and electrons. Okay, the electrons enter the electron transport system here, right? So the electrons are eventually passed on to different enzyme complexes downstream, and the H plus are given off in the periplasmic space. Okay, the electrons enter the electron transport system where they are passed from carrier, like one carrier as in from one enzyme complex to the other enzyme complex, giving up energy at each step. And this energy is actually used to pump these H plus ions or protons inside the mitochondrion from here into the periplasmic, like our intermembrane space. Okay, and more, so this is pumped inside this particular space. More protons are moved out of the mitochondrion by the other enzyme complexes. Okay, eventually as the electrons are passed through, so as the electrons move from complex to complex, their energy is used to pump protons or H plus ions into the space, into this particular intermembrane space. So at the end of the electron transport system, the low energy electrons are joined with protons and oxygen here, forming water or H2O. The energy is now stored in the proton gradient that results from an accumulation of protons between the two mitochondrial membranes here. Okay. The end result is a proton gradient across the inner mitochondrial membrane. So there is a high concentration of protons or H plus ions in this particular space as compared to the um, like inner mitochondrial space here, right? Now what happens here is this particular chemiosmosis is coupled to ATP synthesis. Okay, so what happens is protein. This particular ATP synthase is a protein or enzyme which uses the energy of the proton gradient, which we saw it got created earlier, to make ATP. And the proton gradient is produced as members of the electron transport chain pass the electrons. While passing electrons, they also accept and release H plus. We saw that in the earlier slide. So the concentration of H plus builds up in the intermembrane space. We saw that earlier. And this gradient stores the potential, which is referred to as the protomotive force. And this flow of protons or H plus down the gradient through the rotor and stator part of ATP synthase. ATP synthase is the enzyme. Okay, this is the rotor part and this is the stator part. These are simply the enzyme. If we look at the features of the enzyme, um, and this H plus is flown down across the gradient, and which activates the catalytic sites on the knob portion, and where ADP and inorganic phosphate join to form ATP. So here if we look at this particular picture, so this is the rotor here, this is the ATP synthase enzyme. A rotor within the membrane spins clockwise when H plus flows past through it, down the H plus ion gradient. Like there is the higher concentration in the intermembrane space, still because it is moving against the concentration gradient, so that is why energy is required, right? Otherwise, via simple diffusion, what would have happened is from a lower concentration of H plus ions, like from a higher concentration of H plus ions to a lower concentration of H plus ions, it could easily move through. However, this is being done here with the help of ATP synthase enzyme. And a stator anchored in the membrane holds the knob machinery, and a rod or a stalk, this one here, extends into the knob, also spins, which eventually activates the catalytic sites. And there are three catalytic sites in this particular area, which is the stationary knob here, which joins the inorganic phosphate and ADP molecules to form ATP here. Okay, so these are like very detailed description of what an ATP synthase consists of. Although if we simply go through it, that it is actually helping the rotational motion, actually is helping in moving the H plus ions from the intermembrane space into inside the mitochondria, and this particular static part of the ATP synthase machinery helps in joining the ADP and inorganic phosphate to form ATP molecule. Now we come to plastids. Plastids are actually organelle found, like membrane bound organelles found in plants and algae. And again, it is the site of manufacture and storage of important chemicals. Plastids are definitely the photosynthetic hub, right, where plants actually prepare their own food using chlorophylls. So again, if we talk about plastids in general, it can be chloroplasts, which make sugars using light energy, CO2 and water. There can be chromoplasts as well, which synthesize and store pigments other than chlorophyll. And then there can be amyloplasts as well, which are really non-pigmented. However, they are used in the synthesis and storage of starch granules. So amyloplasts are mostly abundant in stems and seeds. So if we talk about chloroplast, it is a double-layered lipid membrane with a narrow space between intermembrane spaces there. Then there's stroma. This complete part is the stroma, which is the watery liquid containing DNA, ribosomes, and enzymes. Then there's granum. These are the ones, these cylindrical tubes that we see completely. There are stacks of coins if you see that. So these are those coins are thylakoids, and or maybe if we not only coins, we can like kind of related to pancakes as well, stack of pancakes or stack of coins. So each coin is thylakoid and the complete stack is called granum. Okay. And thylakoids are basically flat, hollow membrane covered interconnected discs arranged in stacks, and each stack is connected by channels or tubes here, which are called lamellae. So the connecting tubes between the thylakoids is continuous. Okay. Again, we have photosynthesis, which is divided into two parts: light-dependent reactions, which take place in the thylakoid membranes, these coins, and light-independent reactions, which take place in the stroma here, the watery part, which is outside the thylakoid membranes. Now, if we talk about endoplasmic reticulum and Golgi bodies, endoplasmic reticulum and Golgi bodies are the protein processing and protein secretion unit. So if we see, this is the nucleus here, right? So ribosomes assemble proteins from polypeptides and entering rough ER. So endoplasmic reticulum can be rough or smooth. RER, rough endoplasmic reticulum, or smooth endoplasmic reticulum, SER, depending on whether ribosomes are attached to the ER. If we see ribosomes are attached to the ER, then it is rough endoplasmic reticulum. If ribosomes are not attached, then those are smooth endoplasmic reticulum. Okay, so ribosomes assemble proteins from polypeptides entering rough ER. Proteins move through the rough ER here, right? So this is where proteins are moving through the rough ER, where they are further modified, and then transport vesicles containing the proteins, modified proteins here, the yellow strands here, are pinched off from the rough ER. This is pinching off of the vesicles containing, like transport vesicles containing yellow colored proteins inside. So this is the transport vesicle. And then these transport vesicles eventually fuse with Golgi apparatus or membrane of the Golgi complex, and the proteins are released inside the Golgi complex. So again, further processing of the proteins takes place inside the Golgi complex. And vesicles, finally, these are the vesicles which contain the finished proteins, completely modified proteins, and are pinched off from the Golgi complex here. And eventually, the vesicles travel to the cell membrane, fuses with the cell membrane, and the proteins inside are released off to their target sites. Okay. Then, if we talk about lysosomes, it was discovered by D. Dubé. They are popularly known as suicidal bags because they have roles in autophagy, which is a process where suppose cells under stress or when cells do not have enough nutrition or are starving or they aren't really able to meet their energy requirements, then these suicidal bags come into picture and helps in uh, like cell death. Okay. So what happens is the lumen or the inside the lysosome, it's the pH, the content is extremely acidic and it contains different like macromolecule digesting enzymes like proteases, which digests proteins, cytolytic enzymes to degrade the ingested material. So what is the function? Degradation of ingested food material for delivery through vesicular system, degradation of pathogenic bacteria, degradation of old protein. Okay. And finally, we have the covering of the cell, which is the plasma membrane, consists of phospholipid molecules arranged in two layers. This is why it is called a lipid bilayer. Okay. And if we talk about the phospholipid is made up of a head and two tails here. All right. And heads are actually hydrophilic. They love water. They are polar. And the tails are actually hydrophobic. They are water hating species. And they try to stay. This is the reason that the polar heads are towards the outside, like either outside the cell or towards the cytosolic part, which again is comparatively polar, and the hydrophobic parts or water hating part, or rather water fearing part, are actually kind of protected away from the polar environment, okay, on both the sides. So the bilayer assembles so that the heads interact with the fluid outside and the hydrophobic tails hide in the middle. Okay, so there are many other proteins found in the plasma membrane. There are transport channels, integral proteins, transmembrane proteins, peripheral membrane proteins as well, which have different kinds of functions. Okay. Now we talked about different kinds of organelles, right? Now, if we want to really isolate each of the organelle, then what we usually do is we take the cells or tissues and then homogenize it, and then eventually at different speeds and different time points we centrifuge it. So the centrifugal forces eventually separates out the denser as well as lighter components of the cell, and we can eventually get like separate nuclei, chloroplasts, mitochondria, plasma membranes, ribosomes, etc. Okay. So this is the same thing more or less, where again this is another type of gradient centrifugation, where so since all the individual organelles or the cellular components have different densities, so what happens here is at different density of sucrose, what whenever centrifugation happens, so so suppose if we talk about um, uh, two densities here, A and B, okay, and there are um, suppose two cell components, 1 and 2. So one is denser than the second component. So if one is denser than the second component, then if it can easily pass through the density gradient and settle at the bottom. It will try to sink to the bottom, settle at the bottom as compared to the less denser part, which will try to float in the upper regions. All right. So now when we let's come to the cell cycle. Cell cycle is the sequence of events occurring in an ordered fashion, resulting in cell growth and cell division. Okay. So cell cycle begins at the end of each nuclear division and ends with the beginning of the next. And cell cycle, if we talk about, it is a unit of biological time that defines the life history of the cell. It includes all the significant events ranging from DNA duplication to duplication of cellular organelles and subsequent partitioning of the cytoplasm with proportionate portion of uh, like all the cellular components. Okay. In addition, the process of cell growth, where the cell absorbs nutrients and prepares for its cell division, is also a part of the cell cycle. Right? So, before the cell divides, it has to have all the, like, it should be able to. Uh, it should be able to thrive by itself as well as self replicate later when the time comes, right? So all those requirements, prerequisites have to be there for it to really divide and form a self-replicating entity. Okay. So there are three main cycles that are involved in a complete cell cycle. One is the chromosome cycle, one is the cytoplasmic cycle, and next is the centrosome cycle. So the chromosome cycle involves DNA synthesis that alternates with mitosis, during which the double helical DNA of the cell replicates to form two identical daughter DNA molecules. And then the cytoplasmic cycle comes where the cell grows in size, and during this, the cell accumulates nutrients and growth factors and doubles the contents of the cytoplasm. Eventually, when the cytoplasm divides via cytokinesis, the cytoplasmic contents are equally divided between the two cells. Final cycle is the centrosome cycle, so that where the centrosomes are inherited reliably and duplicated accordingly to form two poles of the mitotic spindle. Okay, and so if we see, this is the complete cell cycle here, and it is divided again into different kinds of phases. Okay, so the very first one, if we talk about, is the gap G0 phase, where the cell is present in a quiescent stage or a resting phase where it is neither dividing nor growing. Okay, so this picture here is showing G1, S, G2, M phase. Okay, all these are preparatory phases and duplication phases, and M is the final dividing phase. Okay, however, uh, sometimes the cells go into a resting phase where it is neither dividing nor growing. So what happens here is this takes place in this particular part, like especially after G1 phase, or usually it enters G1 phase, the cells enter G1 phase when they reach maturity, like in case of muscle cells and nerve cells, but the cells continue to perform their function throughout their lives, right? And in some cases, cells can enter G0 phase from the checkpoint in G1 here due to the lack of any growth factors or nutrients. When basically when the resources become limiting, the cells can even stop dividing or stop synthesizing or duplicating their machinery or whatever components, cellular components, for division. Okay, and sometimes when the resources return or favorable conditions return, the cells have enough resources, then they can eventually again enter the actively dividing phase, like active cell cycle phase, and can come out of the resting phase. Okay, see, that is what is written here. There are some cells like the parenchymal cells of liver and kidneys that enter the G0 phase semi-permanently and can be induced to divide. So, even though G0 phase is often associated with senescence, or aging, the G0 phase is a reversible stage where a cell can enter the cell cycle again to divide. Okay. Then there comes gap 1 phase or G1 phase, which is the part of interphase when cells begin to prepare for cell division. Okay. So cell enters the G1 phase after this division phase, and it is termed as the first gap phase. And here in this particular G1 phase, no DNA synthesis takes place, only RNA gets synthesized and proteins are produced so that the cells can prepare for cell growth. And G1 is considered at the time of resumption where the cell finally picks up. Okay. So when we talk about the gap 1 phase or the G1 phase here, so in G1 phase, what is happening? The proteins are synthesized in preparation, so that the cells get prepared for cell division. Okay, and eventually, this most important part is the transcription of all the three types of RNA which undergo translation or which synthesize proteins, eventually code for proteins to form enzymes or any other proteins necessary for all the other events that will follow in the cell cycle. And duration of the G1 phase is also highly variable. And in some cells, it occupies 50% of the total cell cycle time. Whereas in rapidly dividing cells, the phase is entirely omitted. Okay. And there are checkpoints or at each transition of each phase, at the junction of each phase, where it actually kind of acts as a check to see that if the cell is really ready, cell has all the resources to move on to the next phase, next part or next portion of the cell cycle. Okay. Then you have then you have synthesis or S phase, where events like DNA replication and formation of histone proteins take place. Okay. And all the processes are really tightly regulated, as protein synthesis and replication of DNA require utmost precision, and the histone proteins are produced and other proteins required for newly replicated DNA molecules, for packing of newly replicated DNA molecules. Um, that all those are synthesized during S phase. And the regulatory mechanism of S phase also ensures that the process of DNA synthesis takes place before this dividing M phase here. Okay. And actively expressed genes tend to replicate during the first half of the S phase. Okay, whereas the inactive genes and structural DNA tend to replicate during the later half. So at the end of S phase, each chromosome of the cell has double the amount of DNA with double the set of genes. Okay. Now finally, there is this G2 phase or gap 2 phase here, where the cell collects the nutrients and releases proteins in order to prepare the cell for final division. Okay, or M phase or mitotic phase. So G2 phase is also important, and it checks for DNA damage during replication to ensure that the cell is in proper condition to undergo division. And sometimes this phase might be skipped in some rapidly dividing cells that enter directly into the mitotic phase or M phase, okay, after the DNA replication. So it is, however, an essential phase that checks for mutations as well, checks for DNA damage as well. And DNA repair is actually a crucial step in this gap 2 phase here. And entry of the cell from the G2 phase here to this M phase here um, is regulated by the G2 checkpoint again, this junction and this junction here, where different proteins and complexes are involved. Okay. In case of DNA damage or insufficient nutrients, the cells can remain in the G2 phase and is not really moved on to the M phase or the mitotic phase, dividing phase. Now, finally, the M phase or mitotic phase, most crucial and most dramatic phase, where the most important event is karyokinesis. Karyos meaning nucleus, and division. Nuclear division takes place where chromosomes separate into um, like chromosomal set separates into two sets to form two distinct cells. And this process might differ from one organism to another and even from one cell to the other cell. Okay. And mitosis begins with condensation of chromosomes, which then separate and move towards opposite poles. And a cell entering M phase has 4n concentration of genetic material. However, after M phase, each cell contains 2n concentration of DNA. Again, it has 4 distinct steps: prophase, metaphase here, prophase, metaphase, anaphase, telophase. Finally, cytokinesis occurs. Okay. And then when we talk about the cytokinesis or division of cytoplasm, so it is again divided into two halves, and the cytoplasmic division is actually a little different in case of animal cells and plant cells. So if we talk about the constriction that appears uh, in both the cells and gradually grows and eventually dividing the cells into two halves. Now, however, the what happens here is the contraction of the cell membrane during cytokinesis is brought about by contraction of actin fibers that form a bundle called a contractile ring here, this one here, or this one here. Okay. Now, in case of a plant cell that we talk about, a distinct cell plate is formed here. This is the one, the thicker one in between, and in the middle, and then eventually this cell plate moves towards the periphery, okay, in case of plant cells, and finally leading to two individual daughter cells, okay, or in general two individual progeny cells. However, in case of animal cells, what happens, um, the construction actually starts from the edges and move towards the periphery. Okay, and eventually the furrow deepens inside and two distinct daughter cells are formed. Okay, this is the case in case of animal cells where the construction starts from the periphery and moves towards the middle, whereas from the middle to the periphery, this movement of the cell plate takes place in case of plants. Now, when we talk about the control of cell cycle, so cell cycle at different step is tightly controlled by cell cycle checkpoints, and these are really important to ensure the completion of different steps and repair of DNA damage. And main checkpoints are present at G1S, I mean at each junction or at the transition points of each phases of cell cycle. So what happens if any of these events goes wrong? So dysregulation of cell cycle and control mechanism can give rise to tumor or many other disease conditions. After a certain number of cell division, every cell enters into the G0 phase and stops the cell division. In case of tumor, the cells lose this ability, lose this control mechanism and multiply indefinitely to give rise to a cell mass. Okay. And eventually these cells take up the resources, take up the food resources, take up nutrition, but do not perform any function. Okay. Now let's look at some of the questions here. Um, the genomic organization in prokaryotes is called nucleoid, right? Because it is not really compartmentalized and well defined. Now, which of the following organelle has double membrane? So the double membrane one that we saw is chloroplast. And in case of peroxisomes, peroxisomes are membrane bound packets of oxidative enzymes, mostly found in plants. In plants, they play a variety of roles, including converting fatty acids to sugar and assisting chloroplasts in photorespiration. And in animal cells, peroxisomes protect the cell from its own production of toxic H2O2 or hydrogen peroxide, right? For example, when there are white blood cells which produce H2O2 to kill bacteria or some kind of pathogen. So the oxidative enzymes in peroxisomes eventually are broken down. Um, sorry, in the peroxisomes enzymes that are present inside the peroxisomes, the oxidative enzymes break down H2O2 into water and oxygen. The bond cleaved by lysozyme in bacterial cell wall between N-acetylglucosamine and N-acetylmuramic acid, basically NAG and NAM, in the peptidoglycan layer is beta-1,4 glycosidic bond. The K and O antigens, which acts as surface associated virulence factors, are present in lipopolysaccharide layer, will be the correct answer. So there are certain virulence factors present on the bacterial cell walls. Okay. Um, especially the lipopolysaccharide layer, and they play crucial roles in the virulence of bacteria like Klebsiella pneumoniae, where O antigen is part of the lipopolysaccharide structure and interacts with the host immune responses. And lipopolysaccharide layer is so the lipopolysaccharide layer is the outermost part of the bacterial cell wall. O antigen is exposed on the surface, making it a target for host antibodies. So O antigen uh, has endotoxic activity, protects the bacteria from phagocytosis and bactericidal effects of complement. K antigen affords protection against phagocytosis and antibacterial factors in normal serum. The biomolecule synthesized inside the nucleus is tRNA. All these are proteins which are eventually encoded from mRNA, and the mRNA has to travel out of the nucleus. Okay. The major proteins that cause condensation of eukaryotic chromatin are histones. So if we talk about, this is the double helix, double helical DNA, then eventually it's coiled around these histone complexes, histone octamers. Okay, and the coiling goes on, the compaction goes on to finally give the most compact form of DNA, which is the chromosome. Okay, the phase of Golgi complex that sorts lipids and proteins is called the trans phase. So the final thing is, once the upstream protein, from ER, proteins are coming, incoming transport vesicles are coming. This is the cis phase, and eventually it gets packaged more, modified more. The proteins are modified, and finally there is this maturation phase, or the where the secretory vesicles reach the target sites with the proteins. This is the trans phase or the maturation phase. So the phase of Golgi complex that sorts lipids and proteins is the maturation phase. The orientation of proteins in plasma membrane is predetermined by their orientation on endoplasmic reticular membrane, because that is in general, if you look at all the other membranes, all the other options here, ER membrane is the part where proteins are getting synthesized, right? ER is the synthesized, modified, right? Packaged and modified. So what happens here is most plasma membrane proteins are synthesized on ribosome attached to the rough ER, and while they are being made, they are inserted directly into the ER membrane. And special signal sequences are there, and stop transfer sequences in the protein decide which part of the protein faces the cytosol, which part faces the ER lumen, and the topology is fixed at this stage. Eventually, proteins move from ER to Golgi to plasma membrane via vesicles. So the ER lumen side becomes the extracellular side of the plasma membrane, and the cytosolic side always stays cytosolic. And the protein's orientation remains the same. So histones are synthesized in S phase of protein cell cycle, and it is actually needed to package newly synthesized DNA into nucleosomes. So DNA replication occurs in the S phase. So as to keep the DNA and histone production perfectly matched, histone synthesis is tightly coupled to DNA synthesis. Now, the cells which do not divide once they are differentiated and enter into G0 phase is the lens cells. Okay, so lens cells, specifically the mature fiber cells of ocular lens, terminally gets differentiated and do not eventually divide. So once these cells undergo terminal differentiation, they lose their organelles to become transparent, effectively entering a permanent irreversible G0 phase. While many biological contexts cite neurons or heart muscle cells as the primary examples of non-dividing cells in quiescence or moving into quiescence, lens cells follow the same principle of permanent cell cycle exit upon reaching their mature form or upon differentiation. Okay. But if we talk about lymphocytes in the option, these cells are normally in a quiescent state, but this quiescence can be reversed, because when they encounter when they get a specific antigen or a pathogenic signal, they are stimulated to re-enter the cell cycle or G1 phase and divide rapidly to mount an immune response. Then there are epithelial cells of intestine. These are among the most rapidly dividing cells in the body. They undergo continuous cell division to replace the lining of the gut, which is shed every few days. Then there are smooth muscle cells. So there are skeletal muscles as well as cardiac muscle cells, which are often terminally differentiated. But when we talk about smooth muscle cells, these retain the ability to re-enter the cell cycle and divide in response to injury or physiological signals like repair of blood vessels. All right. So I will end today's tutorial session, and we'll meet the next Sunday.